Thank you very much. Still one presenter after me. Please bear the next 15 minutes. I would like to share the journey of Promethera since inception until today. So here you see Promethera Biosciences at a glance. What is our mission? We are developing cell-based medicinal products for devastating liver indications. How are we doing that? And I will come back to that later. We have 3 therapy technologies for doing that. It is HepaResc, HepaStem, and H2-Stem, as you can see here. And actually the indications that we are targeting are UCD, ACLF, and NASH. I will come back to that in a minute. What is important, our starting material are cadaveric livers, so we had to secure the liver supply. We did that. We made an acquisition of the key assets in April 15, 2016 of Cytonet. And they actually have the agreements, but what is probably even more important, the relationships with almost all of the organ procurement organizations in the United States. So those OPOs are delivering for us now livers, and we do have access to more than 1,000 livers on a yearly basis. Very important if you want to develop indications like NASH, which is a very huge market. You have heard it earlier. It's not only science that you need to work on, but you need to also make sure that commercially you are preparing the company. And we are trying to do both things in parallel. Our IP, we do have in total 14 patent families, covering about 280 patents or patent applications. And the regular term of these patents are between 2026 and 2037, not taking into account potential extensions, legal extensions that we could get. In total, we are about 60 people. So we do have 2 locations. One in the United States, Durham, and one in Belgium. It's close to Louvain-la-Neuve. We also have a broad international shareholdership, and what we also see is that there is more and more interest coming out of the Asian markets. For today, it's more than 15%, 1/5 of our shareholders are Asian investors. So let me come to the 3 technology platforms that we are using. So HepaStem. HepaStem actually is a mesenchymal progenitor stem. So it's not a stem cell anymore. It's already lineage towards the liver. H2-Stem is an hepatobiliary progenitor cell. So that cell type has other properties than the mesenchymal stem cell HepaStem. And finally, we have HepaResc. Those are already fully matured hepatocytes. Here you see actually the liver sourcing supply chain and the production platform. And actually we are mastering the full supply chain. It's an off-the-shelf allogeneic cell therapy cryopreserved product that we are offering. And I think each word here is very important. Liver sourcing I've already discussed. That is secured to the OPO organization network that we have. What is happening then, we are isolating the parenchymal cells, the liver cells. We can, once we have isolated the liver cells, immediately cryopreserve and store. That will give us then the product HepaResc. Remember, those are the mature hepatocytes. Or we can use actually the liver cells as starting material. We can plate them, and subject to the medium that we are using in our manufacturing process, or HepaStem will emerge, or H2-Stem will emerge. We will then expand those cells. For HepaStem, for example, we go to up to 5 population doublings. We cryopreserve the product, we store them. Once there is a patient in a hospital ready for injection, we can ship the product. It can be shipped with a nitrogen dry shipper, and it will be reconstituted at the hospital, where it then will be injected into the patient. And here I think is really the paradigm shift that we could bring in the future, Because actually the only cure for the indications that we're working at, like NASH, and it's more advanced NASH, ACLF, or UCD, is a liver transplant. With a liver transplant, of course, you can only help one patient with one liver. Here, I think, with one liver, potentially, we could help over more than hundreds of patients with one liver, subject to final dosing, of course, of the end product. And this could be really the game changer in the industry. That is our strong belief. Here you see our product pipeline for the three products, HepaStem, H2Stem, and HepaResc. So HepaStem is in the clinic for the acute and chronic liver indication, ACLF. We are in a phase 2A study. I will come back to that later. We've done all the preclinical work for NASH fibrosis, so we do hope to start a clinical trial in NASH in 2018. And we did, that's the historical evolution, we did an UCD indication, we did a phase 1/2 also, but with the acquisition of the assets of Cytonet, we've acquired also HepaResc in UCD, and we decided to push first HepaResc because we have filed HepaResc at Health Canada. I will come back to that also later, so it was much closer to market than HepaStem. Nevertheless, we treated 20 patients with HepaStem in UCD, so that gave us a rich information on data, especially safety data, and that was also allowing us to immediately go into a phase 2A dosing study in ACLF. H2Stem is still in preclinic, so we have very nice data in an FRG mice model, so you see very good engraftment. I will come back to that also in a minute. But we are now working on the next step, and that is making sure that we can expand H2Stem also sufficiently in large quantities. So first, HepaStem, our immunomodulation franchise. And this is actually a very simplified way, but you've heard already a lot about the CAR T cells, and most of the investors today are investing in this space, the CAR T effector cells. Well, we invite the investors to also to look actually to the left-hand side. What we are trying to do, we are trying to cool down the immune system instead of heating up the immune system. And I will come back to that in a minute, how we do believe that we can do that. So we did our homework preclinically. I think you had already a lot of information here, but I will try to do it in a nutshell. So inflammation is vital for host defense against insults. We all know that. These insults can be exogenous or endogenous. So that brings a cascade of cellular and extracellular signals into play that will recruit inflammatory cells. Those inflammatory cells are particularly innate immune cells, such as the macrophages and the dendritic cells. And those cells have a kind of bidirectional function. These cells will engulf the PAMs and the DAMs in a way and present antigens and will produce additional cytokines and chemokines that will lead to the activation then of the T and the B lymphocytes, basically your adaptive immune system. Also through this cascade of signals, your quiescent hepatic stellate cells will be activated into myofibroblast-like cells that will produce a lot of extracellular matrix like collagen 1 and collagen 3. So, and we have now actually did the full analysis of our cell HepaStem, and we have identified a number of anti-inflammatory and anti-fibrotic molecules that could act on these different signals that are produced into the body. More specifically, you see here actually AGF and FGF. You all know that the hepatic growth factor and the fibroblast growth factor are potent mitogens for fibroblasts. So if we can make sure that these hepatocytes will again be cured, that will be very, very helpful. We have also here IDO and PGE2. We know that IDO is a very important enzyme that degrades the essential amino acid tryptophan into so-called kynurenes that are known to exert important immunoregulatory functions. PGA2 also can inhibit actually the proliferation of and the maturation of T lymphocytes, and also the maturation of dendritic cells. Also, AGF has the ability to, and I see that I have only 5 minutes, so I need to speed up. But you see, I have here the full list. We have all the details. People that are interested in that can have much more information, of course. So how are we administrating those cells? It's intravenously through the peripheral vein. So it's a quite easy and simple procedure. We need to make sure, of course, that as it is a systemic infusion, that these cells are passing the lung and go to the homing of the liver and potential inflammatory sites. On the right-hand side, you see here a very nice graph where at first in man was treated. So you see the passage in the lung, but the cells, so they were indium labeled. You see the decrease immediately. You see that they are sticking into the homing into the liver. And what was very interesting, this was a hemophilia patient who had some inflammations in the right ankle and also in the fifth lumbar vertebra. You see that the cells were also going homing into those inflammatory sites. Very promising data clinically that we have also used to go immediately into a phase 2A trial in ACLF. In a nutshell, again, very important markets, ACLF, and our target, we want to push this in Europe and the US. We do have a partner for South Korea, being Life Liver, and we are today also in discussion to find a partner for Japan. But even for Europe and the US, ACLF is already a market of more than 1.5 billion, and that's specifically for the eligible patients that we are targeting. NASH, as we all know, it's the next cancer. It's a multi-billion market. We will focus on severe NASH, F2 and F3 fibrosis, so not early NASH. Very important because there's quite some competition in the field. We are all aware of that, but in the target that we are targeting, the competition is limited. As I mentioned, so we started an open-label phase 2A clinical study in ACLF. We had some issues that we had to tackle. We diligently did that, and we are now actually recruiting patients for the cohort 1B. We have recruited 3 patients. We have treated 2 patients because ACLF is a very severe disease. Up to more than 80% of the patients could die after 28 or within a time frame of 28 days. So from one day to another, the situation of those patients can change dramatically. So unfortunately, we could not treat the first patient. That patient also died. The second patient that has received 2 infusions finally received a liver transplant. So he was saved and could receive a liver transplant. The third patient is doing extremely well, is back at home, and we are now following the parameters, but that patient is back to normal. And as I mentioned, we are now recruiting patients for cohort 1B, and in total 12 patients will be treated. So in NASH, there we have some further animal model that is very encouraging. This is something that we presented at a late breakthrough poster session at AASLD meeting in 2016. Here you see actually the STEM model where the mice are treated so that they develop NASH after 7 weeks, then early fibrosis after 9 weeks, then advanced fibrosis after 12 weeks. So here we took the NASH with early fibrosis, and you see actually the NAS scoring here. That is the NAFLD activity scoring. So the control group, the vehicle that was not treated, had a value of 4, and there are 3 measures actually. It's steatosis, ballooning, and inflammation that is measured. And you see actually with one injection, and you need to bear in mind that all the other products, although it are small molecules, but most of them are daily, so it's within one injection we could decrease the NAS score from 4 to 2.2, so, to 2.2, yes, sorry, so decrease of 45%. Fibrosis-wise, you also see here that there was a significant reduction of 35% in the collagen deposition of the treated mice. So these data are very encouraging. We've repeated this model in advanced fibrosis. That data is not public yet. Again, happy to share with people who could be interested, but basically we saw the same effect as we saw in this model. Quickly, perhaps, on H2-stem, very simple. We treated FRG mouse. Basically what is happening, it's you make the mice immunodeficient, and also you make sure that the actual mice hepatocytes are dying. You're then injecting normally mature hepatocytes. We've injected H2-stem, and you see actually how nicely these cells were repopulated into the liver. We followed them up to 8 weeks, 2 months, and here we stained them for albumin, arginase, and OTC. So quite impressive. You see here the engraftment, but I need to speed up, sorry. Health Canada, and this is basically my almost my last slide. I mentioned that we filed at Health Canada for this product. To be very transparent, so it was refused market authorization for Europe, but there was a divergent opinion letter, so 12 out of the 28 member states do thought that there was value, and we are supported now also. There has been a publication, happy to share that with you, and you see the key results, 7 patients, that there was really an effect that was shown. Very quickly, very international shareholder base, a seasoned management team, and this is really my last slide. So we do hope that we can finalize the ACLF study beginning of 2018 and can start also in the clinic a NASH study. Thank you very much.